That gives us one more chapter that we can finish up next week pretty easily and still have some time to do, say, a lab activity and some other review stuff. And again, the test is 2 weeks from today. 2 weeks from today is your third test in the test over chapters 8, 9, and 10. So chapter 9 is focused on basically how you get energy from the food that you eat. All right? And what's the major organic molecule we're going to be starting with in this process? Glucose. Good. Have you seen this diagram before?
Yes.
Yes, you've seen it before. But I decided to make it the first slide today, since it was one of the last slides we ended with last time. And that way, in this bit of lecturing, you will see every phase. So there's really 4 general phases to cellular respiration. And we're going to start with glycolysis, which happens where? Outside the mitochondrion, out in the cytoplasm. That's right. And basically we are taking glucose and breaking it down into a chemical called pyruvate. All right? Now you'll be given these diagrams. I won't tell you on the test that this diagram is glycolysis. That's a question you might have to answer and tell me. But knowing how to spell pyruvate or even what its chemical structure is, that's not quite what we're focused on here. Glucose is being split up into 2 smaller, simpler, lower energy molecules. And in that process, what are we getting out? What's the benefit for a cell for doing this? ATP production is part of it. Now remember, we had to put a little ATP in, but we get more ATP out, so we have a net gain of ATP. And then what is this? It's an electron carrier, NAD+, picking up electrons, and now it's in a state where it has high energy electrons that it's ready to deliver somewhere. Where is it going to deliver these electrons? Tune in to find out. So, I just had some of this up here just as reference for later for you, and a few terms that I want you to be aware of. For instance, this idea of aerobic pathways. What does aerobic refer to? It refers to oxygen, the presence of oxygen. That's right. So, we are assuming that the cell has access to oxygen, O2 molecules. That's the assumption we're making for the rest of these phases. If it doesn't have oxygen present, at the very end today, we're going to talk about what happens instead of all this stuff. Now, as it mentions up here on the board, we have broken glucose down into pyruvate, but there's still a lot to squeeze out of that pyruvate. So that's what takes us to the second phase, called pyruvate oxidation. What does oxidation mean? Removing electrons, losing electrons. So we're going to keep oxidizing, keep breaking down pyruvate. So this is the chemical that we ended glycolysis with, okay? So one phase ends with some product, and here we are starting with the previous product. This is pyruvate right here. And what do you think this represents? Not just the mitochondrion, you're right. Both the outer and inner membranes of mitochondria. That's right. So there is going to be a transport protein that is responsible for bringing pyruvate in to the mitochondrion. And there is actually, while there isn't a whole lot on this diagram, there are a few things that I am not particularly focused on. I don't really care about this enzyme here that is going to further break down pyruvate. You don't have to know about really most of the enzymes in this process. But essentially, pyruvate, this 3-carbon molecule, is going to be stripped down a little bit more into this very oddly named chemical called acetyl-CoA, which has only 2 carbon atoms and then what we call a coenzyme bonded to it. And we're not really getting into coenzymes. These are chemicals that help processes along. But there are 2 things I really want to stress here in this phase. One, what are we doing again? Picking up more electrons with our electron carrier. We are just collecting electrons every chance we get. Collecting spare parts for using later. And I mentioned that we strip down or break down pyruvate. Part of that process is taking this carbon and 2 oxygens, and look at what we're pulling off. Carbon dioxide. Why is that kind of interesting? What do you do with that? It's a waste product you're breathing out, that you're getting rid of by exhaling. So why do you breathe out carbon dioxide? Well, one reason is that some of it is generated at this stage. So this is carbon dioxide coming out of the reaction as we are stripping this molecule down to get it all the good stuff. And some of that good stuff this molecule has inside it, we are taking away in the form of electrons. And then we end up with a 2-carbon molecule as an end product. A pretty quick phase. Not a whole lot happening, but where does this happen? Inside the mitochondria. Do you remember what we call the very inside region, the space inside? The matrix. This is the matrix where this happens. Inside the outer membrane, inside the inner membrane, all the way into the matrix. Okay, so we have this acetyl-CoA now that we have produced in the matrix, and we've picked up, some more electrons from our electron carriers. By the way, and this is where the AP Bio style questions start coming in. Think about, let's look here real quick, go back to this first diagram here. This is where we left off. Okay, that's where we left off. And then we enter into this phase right here. How many times is this going to happen for every glucose molecule that we start with? Twice. 2 times. Stop answering my questions before I finish them. That's right. So this is a pyruvate. 1 pyruvate out of the 2 that we got from glucose. So for every glucose molecule, you're actually getting 2 molecules of carbon dioxide. You're filling up 2 of these electron carriers called NAD+, to make 2 NADH. And you're getting 2 acetyl-CoAs for every 1 glucose you start with. So there's a little bit of accounting that we do with some of this stuff. But if you have these diagrams in front of you, you should be able to follow them and make sure the numbers check out. Okay, so this acetyl-CoA. Again, I know these names are weird, but They'll be written on these diagrams, so you don't have to memorize them. Just understand that that is the name for this final product here. It is in the matrix. And also in the matrix, it is going to enter into what we call the citric acid cycle. This is the third phase of cellular respiration, assuming oxygen is present. Now, this is the one that looks super duper scary, and it's one that, as your book discusses it, you may start feeling a little lost. But I want to try to simplify it, boil it down to the basics. So here at the top we have acetyl-CoA. So this is what we just made. It is going to enter into a cycle of chemical reactions that actually kind of end with a beginning reactant here. So acetyl-CoA, which has 2 carbons, is going to bond to this oxaloacetate, which has 4 carbons, and make a 6-carbon chemical called citrate, also known as citric acid, which is where the name citric acid cycle comes from.
You know what's in like sweets?
They do put citric acid into some products, and I don't know if it's for flavor or like probably not a preservative. It probably gives it like a sour flavor in some cases. So in some candies they add a little sour flavor too, along with all the sugar. Now, I am not asking for you to memorize what things bond together and what they turn into as you go around this cycle. Again, you'll probably see this exact thing just like this, maybe without the name citric acid cycle written in it. And I will ask you questions based on what you're seeing here. And I might ask you something like, as isocitrate is converted to alpha ketoglutarate, what is happening? And you need to be able to find it and tell me what is occurring specifically there. But essentially, as we're moving from one chemical to the next, each step driven by a specific enzyme, okay? As we are moving from one step to the next, there are a few points where important things are happening. For instance, what is happening right here? We are picking up more electrons to carry away to the final step, where we're going to find out why we've been picking up electrons this whole time. What does this indicate? Carbon dioxide production as a waste product. And you can actually go through. There are 1, 2, 3, 4, 5, 6 carbon atoms here. Okay? We lose a carbon dioxide. How many carbon atoms does alpha ketoglutarate have? 5. So we are actually degrading it down. We pull out another carbon here, continuing to strip down these chemicals, and as a result, we are able to pick up more electrons. In this step, what are we actually producing? ATP! We're making some energy. Good job, Ian. This is a little weird. FAD becoming FADH2. This is simply a what? It's different from another one we talked about, but it's simply an electron carrier. FAD and NAD are both types of electron carriers, and we are picking up electrons here in the form of NAD, here in the form of NAD, but here FAD is a different electron carrier. I don't know evolutionarily why you have a different one at this step, but the cell is employing the help of a different electron carrier, and we're carrying electrons away with it. And then there is some change from one to the next one last electron carrier is going to pick up electrons, and then we end up at the chemical that bonds to the next acetyl-CoA molecule that's available, and you repeat the process. So, and remember, this is for one acetyl-CoA. So, for every glucose molecule, how many times does the citric acid cycle turn? Twice. One glucose molecule would be broken into 2 pyruvate, each converted into an acetyl-CoA. So you're going to get this cycle to turn twice for every glucose molecule. You produce a lot of carbon dioxide in this process. That's where the carbon dioxide comes from that you're breathing out. It comes from the breakdown of these chemicals that originated from glucose. And we are carrying away a lot of electrons in this process. And the reason we have been catching electrons during glycolysis and during the pyruvate oxidation phase and during the citric acid cycle is because they're all going to be used in the last step that I'm going to go over in 1 second. Before I do, there is another name for this cycle. Sometimes it is called the Krebs cycle. K-R-E-B-S. Krebs cycle. I think that's apostrophe S for possessive. I believe Krebs is the scientist who first discovered or worked out what was going on in this cycle. So if your book, or if I ever say Krebs cycle, it's the same thing as citric acid cycle. So, the last step of cellular respiration, if oxygen is present, is what your book refers to as oxidative phosphorylation. But a lot of times I will just say electron transport chain, because that's kind of a major half of this process, and we'll get to the finale here in a second. Now, what is this thing right here? That's a membrane. It's the inner membrane of the mitochondrion. Okay? So there's an outer membrane somewhere up there, or out there. This is the space between the outer and the inner membrane that we call like the intracellular space. And then this inside the inner membrane is simply the matrix. Okay? So this is where we've been having the pyruvate oxidation and the citric acid cycle, basically capturing a lot of electrons in the form of electron carriers. Okay? So most of your electron carriers are going to be picking up electrons in this space. And check it out! Here they are! Here they are! NADH and FADH2. So, what exactly are we looking at? In purple, what are all of these purple structures associated with and within this membrane? They are proteins. That's what the electron transport chain is. It is a series of proteins. And some of these are specifically transport proteins that carry materials across a membrane. And we're going to talk about that. But essentially, all this NADH and all this FADH, that leading up to this point, I've been like, hey, look, NAD got an electron, FAD got an electron. All those electron carriers are carrying electrons to these proteins, specifically kind of the first proteins in the chain. So NADH, FADH, they're carrying electrons and they're dropping these electrons off. And that's what you see right here. Look, it's being oxidized back into NAD+. So it has an electron, doesn't have an electron. Where did the electron go? It went into this chain of proteins, this series of proteins. Now, these electrons are going to hop from one protein to the next. Notice FAD2 drops its electrons off at a different protein, but that's not terribly important for our purposes. But the electrons that are dropped off here are going to move from one protein to the next. Okay? Now, at every jump they make, a little energy is given off. And with that energy, the cell does a little work. And what is that work? It takes hydrogen ions from a low up to a high concentration. See all these H pluses that are written up here? This is in a very acidic, low pH environment. And the cell is intentionally moving hydrogen out of the matrix across the inner membrane to the other side. So we are intentionally creating a very I'm kind of lost the word here. We're creating a concentration gradient. We're creating a concentration gradient. A pretty significant one, I guess is the word I was looking for. So the energy from these electrons hopping from one protein to the next is the energy these proteins use to move hydrogen against the gradient. Now that's going to be used in the very last step here. There is a very special protein in the inner membrane of the mitochondrium, and it is called ATP synthase. What does that ACE ending tell you? It's an enzyme. It's an enzyme. Now, this ATP synthase allows all these hydrogen ions to flow back through from high to low. So we pump them from low concentration up to a high concentration through active transport. And now they're going to diffuse back down. They're going to diffuse back down. It's almost like your cells have created a waterfall, except instead of water falling and maybe turning a turbine like we see in a hydroelectric dam, it's not water, it's hydrogen ions that are pouring through this enzyme and giving the energy that this enzyme needs to add a phosphate group onto an ADP molecule to make ATP. And you might think, oh, we made a little ATP. It's a lot of ATP. It is somewhere over 30 molecules of ATP for every glucose molecule. Yes, it's true. I know you're all shocked and in awe of that number. So you think about like glycolysis, we made a profit of 2 ATP molecules. You know, wow, big deal. Here we're making more than 30 ATP molecules for every glucose we start with in glycolysis. By the time you get here, it's over 30. There's actually some debate about the exact number. So I just say over 30. The point is, a lot. a lot of ATP is produced. Now this process of hydrogen kind of moving through from high to low and driving this whole ATP production, we call this chemiosmosis, which is, it's a word, but not a word I use very often. This is the big deal. This is what keeps you alive. This is what gives you the energy to do all the things you need to do. The ATP production from ATP synthase. It is driven by hydrogen ions moving through it from high to low. It actually, like a turbine, this enzyme literally spins as these ions are moving through it. And that mechanical energy somehow allows for this process to occur. How did we get all these hydrogen ions up here? The active transport that needed the energy that we got from the electrons that our electron carriers dropped off. Where did they get all of their electrons? Where did these come from? All the other steps we just discussed beforehand, where we stole them from basically glucose being broken down. Now there's one last point I want to make, and it relates to the point I started with, which is this only happens if you have oxygen available. Because, and I probably should have said this earlier, but I wanted to tell you about the end part here. These electrons that are hopping from protein to protein, when they get to the last protein, they need to be carried away. They're done. We have used them. The next electrons are trying to jump into the next place. So, based on this diagram, what takes the electrons away from this electron transport chain when they are done being used by the chain? That's where oxygen comes in. You breathe so that you can get oxygen into the matrix of your mitochondria so that it's there to take electrons out of your electron transport chain. When oxygen grabs them, it actually converts the oxygen into water using some of these hydrogen ions that are available in the matrix, which further makes this a lower concentration versus a higher concentration. So you are actually turning oxygen into water in this process of grabbing the electron at the very end. We call oxygen the final electron acceptor. It is the last thing in this process that takes the electron. If you stop getting oxygen, it stops being available in the matrix, and these electrons stop leaving the transport chain and hopping onto them. And if the electrons stop leaving, they just stay in these proteins, and no more new high-energy electrons can be dropped off. And if that stops happening, you can't pump any more hydrogen across. And if you can't pump any more hydrogen across, it stops flowing through. And if it stops flowing through, you don't make like 30-something ATP molecules. And instead, the only ATP you have is like one molecule you made in the citric acid cycle, which actually wouldn't even happen because the whole process shuts down all the way back to glycolysis. Glycolysis is the only thing that will happen if you run out of oxygen. And you can run out of oxygen pretty fast. Just hold your breath and start feeling those electron transport chains shutting down.
Is that when you get like lightheaded?
You know, the lightheadedness that sometimes can be caused by kind of blood pressure changes and all. Like a lot of times if you like squat or kneel down and then you stand up real fast or get out of bed real fast, a lot of times that's just blood flow. But that is blood flow carrying oxygen, so yeah, I mean, it would cause a bit of a lightheadedness. Now those are just your body's ways of like, you know, indicating to you that your oxygen supply somewhere is not enough. So that is how you go from glucose to all the ATP you need. Those 4 steps. And again, I would give you this diagram probably without these kind of terms labeled, but pretty much everything else. I might just ask you, what is this? Electron transport chain. What's the final electron acceptor? Oxygen. Good. And other questions that I could ask about that. Okay, so quickly, I know we've been doing this for a little while, but we're almost done. There is, to finish up chapter 9, there is this concept of fermentation. This is what happens when you don't have enough oxygen or when you run out of oxygen. Basically, nothing in the mitochondrion will happen. The pyruvate oxidation, the Krebs cycle or the citric acid cycle, that electron transport chain we just looked at, all of that shuts down when oxygen is used up. So what happens when you have fermentation, which means no O2, no oxygen? Fermentation can happen. Now there's 2 ways this can happen. Alcohol fermentation happens in yeast cells and in some other bacteria, but not in you. And essentially, glucose is going to be broken down into pyruvate molecules through glycolysis. But then instead of that pyruvate going into the mitochondrion and all of the things we just talked about happening, it is essentially going to be converted into what we call ethanol, which is the alcohol in alcoholic beverages. Now, you still get a profit of 2 ATP molecules. You still make a little ATP. But the problem is that you really don't make any other energy. The goal of fermentation then, basically at one step, you're able to kind of oxidize this NAD, and then it is available for glycolysis again. So you're kind of, no pun intended here, but it's kind of spinning the wheels right here. You're getting a little ATP, a little bit of energy, but all the big processes that provide all the big time energy are shut down. So all you're doing is creating kind of this waste product, and in the process of doing that, you're freeing up your electron carriers so that they can at least participate in glycolysis again. We do this intentionally. We feed yeast sugar, we seal it up, the oxygen runs out, and the yeast makes ethanol. That is how you make beer and wine and all kinds of alcohol. In fact, the big difference between a lot of types of alcohol is just where you get where you get the glucose from. So in the case of, say, whiskey, we get our glucose source from corn, but for rum, it's what? Sugar cane. It's sugar cane. Sometimes they flavor with coconut. Vodka, they get the glucose from potatoes. Wine, the glucose comes from grapes. Tequila is from the agave cactus fruit. So that is the process that we use to make alcoholic beverages, which is the production of ethanol, essentially. This is not what we do. And that's good, because you go into fermentation after you run for a few minutes. You start using up pretty much all the oxygen. You start breathing hard to get as much oxygen replenished as you can. But you will have cells that begin fermenting not too long into exercise. And it's good that your cells don't make ethanol, because you would become intoxicated from that exercise. Instead, animals tend to undergo what we call lactic acid fermentation, which is essentially the same thing, but with a different end product that is not ethanol and not poisonous to us. Lactic acid, that is going to be the end product. But again, the whole point of fermentation is just to make sure glycolysis can keep happening. So at least you get a little bit of energy. Next week, we will talk about photosynthesis and how plants get us the glucose. It would be very much worth your time to look back through these diagrams, start with glucose, and just follow the path all the way to the end